My name is Rachael Onyegbule, and my research project was on investigating hydrogen peroxide-induced oxidative stress, antioxidant response, and cytotoxicity in shy neuroblastoma cells. Oxidative stress plays a central role in neurodegenerative diseases such as Parkinson's and Alzheimer's. It occurs when there is an imbalance between the production of reactive oxygen species, also known as ROS, and the cell's ability to neutralize them. Cells in the central nervous system are particularly vulnerable to oxidative damage because they have high oxygen demands and a relatively weak antioxidant defense system. To study this in a controlled setting in the lab, researchers often use shy human neuroblastoma cells because these cells express neuronal markers and respond to oxidative stress in predictable ways, making them excellent in vitro studying for neurodegeneration. I hypothesized that the exposure of shy cells to hydrogen peroxide would increase a dose-dependent increase in intracellular reactive oxygen species and upregulate the expression of the antioxidant enzyme, that's GPX1, and reduce cellular viability. To test this, I had three objectives. First was to assess cell viability using the MTT assay, and the second was to quantify intracellular ROS using the DCF-DA assay with normalization to cell viability. Third, it was to measure GPX1 gene expression using quantitative PCR. So for the methods, shy cells were cultured in standard cell culture conditions. Fresh hydrogen peroxide solutions were prepared immediately before each experiment at concentrations ranging from 0 to 1,000 micromolar. For the MTT and ROS assays, cells were seeded into 296 well plates and treated for 2 hours. For the qPCR analysis, cells were seeded in a 12 well plate and treated also for 2 hours with concentrations of 0, 25, 100, and 200 micromolar hydrogen peroxide. RNA was extracted using the ARM kit, and cDNA was synthesized using the iScript kit. Quantitative PCR was then performed using primers for GPX1 and the housekeeping gene GAPDH. MTT assay revealed a clear dose-dependent decrease in cell viability at low concentrations of 10 to 50 micromolar. Cell viability remained above 80% with no significant difference from the control. A significant reduction was first observed at 75 micromolar, with viability dropping to approximately 57%. At 100 micromolar, the viability decreased to about 51%, representing approximately 50% cell death. At 200 micromolar, the viability was reduced to 33%, and at 1,000 micromolar, the viability dropped to only 12%, which was comparable to the positive control. The DCF-DA assay showed that intracellular ROS levels increased with increasing hydrogen peroxide concentration. After normalizing the ROS signal to cell viability to account for cell loss, I observed a progressive increase in ROS per viable cell. Significant increases were first observed at 75 micromolar, and peak ROS production occurred at 200 micromolar, reaching 410 compared to 100 in the control group. At 300 micromolar, ROS levels decreased slightly. which may reflect reduced metabolic activity in the remaining stressed cells. And at 1,000 micromolar, ROS levels rose again, though this may also be influenced by the low viability at this extremely high concentration. And for the gene expression, quantitative PCR analysis revealed that GPX1 expression was upregulated following hydrogen peroxide treatment. At 50 micromolar, expression increased to about twofold, but this was not statistically significant. At 100 micromolar, expression increased significantly to approximately 6.3-fold above the control. At 200 micromolar, expression reached approximately 6.6-fold above the control. The large variability observed at 100 micromolar suggests that the concentration represents a transitional point where some cells mount a strong antioxidant response, while some others begin to undergo cell death. In conclusion, the study successfully established an in vitro model of oxidative stress in Schillei cells. Hydrogen peroxide exposure induced a dose-dependent decrease in cell viability and a dose-dependent increase in intracellular ROS per viable cell. Importantly, the GPX1 expression was upregulated at the high concentrations of hydrogen peroxide, with approximately over six-fold observed at both the high concentrations. This indicates that Schillei cells mount an adaptive antioxidant response to oxidative stress. These findings support the hypothesis that oxidative stress triggers an adaptive antioxidant response in neuronal cells, and it provides a useful model for studying cellular defense mechanisms against oxidative injury. Thank you for listening.